Yo, welcome. Welcome to the Syndicate. So I'm going to peel back the curtain on a new project that we're working on. It's like new to you, but old to me. And if you don't know who I am, my name is Dr. James Utley. I am your biohacker extraordinaire. So anyway, I want to tell you about a project called NucleoGenesis that I've been working on. Okay, we're moving on to NucleoGenesis here. So NucleoGenesis, let's start at the top. The wildest idea is we're trying to unlock immortality by developing a clone from a nucleus that's harvested from dried blood. Now, that sounds crazy, and indeed it is, but that's what we do here. But now let's break it down here. So the Nucleogenesis project is a research program that I'm developing here, right? And it has two dual systems. One is to preserve and digitally reconstruct a human leukocyte nucleus. It's probably specifically a T cell nucleus. So now how do we do that? We use a chemically treated cellulose card and we store it in temperatures to try to preserve it. So we try to preserve the card and then we stored it at a high monitor temperature or a highly monitored temperature, and we pair that with some ML AI based software that we have broken down as the sampler, the builder, the reconstructor, and the synthesizer, right? Or the synth, right? And so that's how we convert preserved samples of nuclear material and transform them into digital nuclear files. That's a nucleus, right? This method it removes the need for cryogenic storage and is like the ultimate thing it is designed to survive natural disasters or system failures or authoritarian governments or just the destruction of Earth in general. And it's the project, it uses a two-phased approach, which is a top-down reconstruction, which is from the material, and then also we develop a bottom-up digital synthesis, and that gives us a real model. And so if you don't believe me, I'll show you. This is the reconstruction module here that is now in the window. And what we do is we take all the data, all the required data, and we build a digital model. And that digital model sits here. It sits here in this window for you to look at. And now we're going to add the membrane. So we add the membrane now, and all of that stuff in the middle is nuclear material that is from data files that has been digitally synthesized to then construct a nucleus, right? And again, this has the goal is to create a reliable, low maintenance platform for long term nuclear storage and the future of synthetic reconstruction for scientific and medical use. So you could be able to take your own nucleus from a white cell, then be able to preserve it in two ways. You're preserving it physically through the dried blood spot card that has been chemically treated to preserve the nucleus. That's not the red cells, it's usually. And then, or on top of that, is you are also generating a digital file that is trying to be able to provide information to synthesize that nucleus downstream. Now, that is the essence, so that you could, if you needed to clone your whole body, you could. If you needed to clone for some tissues, you could. If you needed to take your card and send it on a vessel into deep space, it would survive. And it is in the essence of preserving something, and then building it in the future. So it's decrypt, collect now, decrypt later, collect now, decrypt later. This is all beginning. We have been researching this deep, and I continue to do so. Please help me out, support the project, and